Hello everyone. Have you ever wondered about the physics of drop coalescence? It's artistically beautiful, but do you know that it has a great scientific importance in research or maybe in nature? Especially currently, we're living in a difficult time where COVID-19 devastated our personal life, social life, and I can make a good connection in this research to the COVID-19, I guess. We still don't know the virus fusion mechanism, but I think we can look at it from another perspective. I'll tell you why. Before that, let's dive into the physics of drop coalescence. I hope you will enjoy it. So, drop coalescence, uh, think about it, two drops coming close to each other, and upon touching them, they may form a bridge, liquid bridge between them. Once there is a stable bridge formation, then the drops coalesce to each other and it propagates and eventually completes the coalescence. So here you will see a very fast phenomenon of drop coalescence. You see here it's 800 times slower. That means you will not be able to see your bare eyes. Let's see the movie. So you saw here is a three-dimensional coalescence. I'll show you now a two-dimensional coalescence. So here, two drops are similar, contain similar kind of liquids. One contains the dye, and another just transparent dye water. And these two drops are sandwiched between two glass slides. And if you see the movie here, you will see that it forms a sharp boundary between them. So two liquids, this one and this one, does not invade to each other, and the later drop shape becomes slower compared to the three-dimensional coalescence. And if we see the same coalescence with particle added to it, you should be able to see how fluid moves at the beginning and at the end. So from the PIV analysis, we saw something very interesting for 2D coalescence. Here, on the very left bottom image, you see at the onset of coalescence, something very remarkable we are able to see from the PIV. So these are the streamlines, and if you see that there is no gradient in the perpendicular direction of the streamlines, meaning that the fluid layers, there is no friction between fluid layers coming towards the bridge region or towards the other drops. That's interesting. So whole drops in the bulk region is not uh, resisted by any viscosity. It is happening in the bridge region and mixing interface region. So we divide the bridge region in two parts. We call this region as the mixing interface region, and this region we call a bridge region. So at the mixing interface region, you see particles are coming towards the other drop and then deviates perpendicular in this direction, so radially outward. And you see, if you see this component, the y direction of the r direction of the velocity component, you see the velocity gradient is higher compared to in this region. You see the velocity is higher compared to this here. That means there is a gradient here, but the gradient is higher than here. That means there is a viscous resistance here, and also there is a viscous resistance here. So we observe three things here. One is the bulk drops in the coalescence. Bulk drops is inertial. The fluid motion in the bulk drops is inertial, and fluid motion in the bridge region and mixing interface region is viscous. So there is a resistance coming from the viscous occurring in the bridge region and mixing interface region. So this observation we applied in 3D coalescence. We formulated a scaling law to validate if our understanding is true or not. So in the next slide, you see here we conducted 4 experiments with 4 different kind of viscosity of the liquid, and you see our theoretical prediction marked by the solid line and the experimental data match quite well. That means what I told you from the 2D coalescence is true. I mean, the physics is true for the 3D coalescence as well. So from this experiment, we find out that there are 2 different dynamics happening in coalescence: the early dynamics and the later dynamics. So in the early dynamics, fluids are uh, in the early dynamics, the whole coalescence phenomena governed by the Ohn-Sugg number. And later dynamics, you really can change the dynamics by using different kind of imposing different kind of viscosity, or I mean another kind of resistance. It could be using a different surface, as we have shown here in 2D coalescence. We sandwich 2 droplets in the between the 2 surface, and at the later dynamics, that added some resistance, and these change the dynamics as a whole. And also you can do, like, you can add some other things to tweak initial dynamics, the viscous dynamics. I'm not going into detail, but we published these things in our paper. See one more example here. So this is this is a 3-dimensional coalescence, and this is 2-dimensional coalescence. As I said, I think I already explained this part, but let's talk about this scenario. So this drops contains surfactant, and this drops does not contain surfactant, both having DI water. So this drop has a higher del P. That means the pressure when it forms a bridge, pressure between bulk and bridge is higher here compared to the here. That means in early dynamics, this pressure is higher, so it will form a liquid column. So once this form a liquid column, it will sort of invade to the other liquid. And in the later dynamics, when other resistance dominates, like this is a surfactant, surface tension here is higher compared to here, so we'll be able to see the Marangoni effect in the later dynamics, and we can see a nice shape coming from the later dynamics. See, at the first, liquid column is invading to the other drops, and then the surface pulling the other liquid in this direction. So by working on 2 different dynamics, there are a lot of materials you can generate, and you can work on just two drop coalescence in different size. Maybe you can work on many coalescence, and you can generate different kind of shapes. So I would say this study is very fundamental in any application where 2 drops merging occurs. So we published our work in paper in physics of fluids. If you're interested in this research and know a lot more, I would request you to go through this research. And if you really like it, please let me know why you liked it. And if you have any concern questions, please let me know, and you can share our research. I hope this research will fundamentally change in many areas. As I said in the beginning, we are experiencing a tough time, the COVID-19, and I believe that we don't understand fully the virus fusion mechanism inside the cell. I think we need to incorporate this drop coalescence phenomena, this biophysical phenomena in the cell biology research. For example, you can think about it: the cell membrane and the protein on the virus carrier. So there could have any propensity that those two things, the cell membrane and this one is the protein, propagates this, initiates this coalescence, and so definitely there is a early dynamics and later dynamics playing a role here, and probably the size matters of the virus sphere, and also maybe the constituent like the protein. It can form a stable bridge. It may not form a stable bridge. It can bend the bridge region, and it can um, move back to the original drops and does not propagate the coalescence. So definitely, if you take this research in the cell biology research, and I'm very confident that we will be able to make a very good conclusion about a drop, about a virus or other kind of other kind of stuffs in infusion mechanism inside the cell. So I hope you liked it. Thank you very much for your attention.